Good evening, Amita. Good evening, Vanshika. Okay, so I'm Rahul Saha, and in the last class we had talked about the specificities regarding the follicular phase, right? So in today's class, I'll just show you one of the negative feedback loops, right, that pertain to this specific phase, and this is extremely necessary to know and to understand the negative feedback loop mechanism, okay, associated with the or associated with each phase of the menstrual cycle. So if you're unaware of negative feedback looping, okay, the rule is, as the name suggests, negative feedback loop. I don't know whether you have ever studied about constructing a negative feedback loop in your endocrinology chapter in or the last chapter, chemical coordination, class 11th. So what happens in this is a way, like our body has a specific way of regulating every specific pathway that is perpetuating or that is occurring. Okay, none of the pathways are incessantly happening, right? So if a hormone is being produced, there will be a time when the hormone doesn't need to be produced anymore, and at that point in time you need to, you can say, down regulate the amount of the hormone, otherwise it will cause severe side effect. Okay, same way in glycolysis, the glycolysis cannot go on incessantly, ceaselessly, because then you will be making too much of, you can say, NADH molecule in the cell. And I told you, too much of NADH, NAD plus ratio, if it is too high, then you will be producing too many reactive oxygen species. Again, that will be detrimental for the cell, that I had talked to you about in few classes before. That I had shown you glutathione pathway, superoxide dismutase, superoxide radical and all, right? Then I had also shown you about, you can say, peroxidases, right? All those things that we had talked about. So glutathione, peroxidase, glutathione reductase. So those things will be, you can say, they'll go on to overburn, right? They will be upregulated uselessly, and we will be uselessly producing too many, you can say, oxidized GSSG form of glutathione. So here negative feedback loop in menstrual cycle means that if at all too much estradiol, that is estrogen only, or too much progesterone is produced, okay, then their own level, okay, their level will go, and because they will be in blood, right, their endocrine hormones. So there are three pathways: autocrine, paracrine, endocrine. So endocrine hormones, they'll be in blood, they are all steroidal hormones only. So they'll go to their target organ and specifically inhibit that target organ, okay, that target organ from secreting any more FSH or LH, because FSH or LH secreted by the target organ, like the pituitary gland or the target gland, will enable the synthesis of estradiol and progesterone, right? So that will occur. So that does not need to be synthesized. So that is a way of negative feedback looping. Okay. So what happens is, I'll draw the negative feedback loop for you regarding the follicular phase. So keep in mind, this is the negative feedback loop pertaining to the follicular phase only. Okay, so we have hypothalamus. Let's see. We have hypothalamus that would secrete GnRH, right? As per the current nomenclature, N is written as capital, right? Not small anymore. Gonadotropin releasing hormone that would go on and bind to the adenohypophysis or the anterior pituitary gland, right? You can directly call it anterior pituitary gland. That would enable or stimulate the pituitary gland to produce follicular, these are the protein hormones, follicular stimulatory hormone and luteinizing hormone. These two hormones would be secreted. I told you that the receptors for FSH, few of the developing tertiary follicles would express the receptor for FSH, meaning that the granulosa cells will be expressing FSH receptors, whereas the theca interna, right? Theca interna cells would express, you can say, the LH receptor, right? This will produce androgen, androstenedione, then testosterone, okay? And this will lead to, okay, formation of inhibin, okay? Inhibin, also androgen. When this will go through this will also produce aromatase that will convert this androgen into 17 beta estradiol. Okay, simply you can call it estrogen only. Okay, so what happens is the follicular phase, just understand now the specificities. The follicular phase, it marks the beginning of a new cycle. Okay, as the follicles, which are nothing but oocytes, which are then surrounded by the zona pellucida, then surrounded by the granulosa cells, multiple layers, 3 layers at least, and then membrana granulosa, then followed by the theca interna and theca externa, the stromal cell or connective tissue. Membrana granulosa acts as the basal, you can see the basement membrane, which is composed of basal lamina and reticular lamina, connecting the epithelial type granulosa cells to the connective tissue type stromal cells, right? Now, at the start of this new cycle, there is little ovarian hormone production, and the follicle begins to develop independently of gonadotropins or ovarian steroidal hormones. Now, due to the low steroid hormone, meaning that low estradiol, okay, and low inhibin levels, there is a little negative feedback loop. Okay, little negative feedback loop means this will be produced in very less amount, so it will still bind to anterior pituitary gland. Minus means it will inhibit, okay, FSH only. Inhibin only inhibits FSH. Okay, always know that it will inhibit, you can say, anterior pituitary gland to secrete FSH. Okay, FSH only means it will inhibit the secretion of FSH only, not LH from anterior pituitary gland, and it will not bind to hypothalamus again. So these are the two specificities regarding inhibin. Means first one is it will only go and bind to the anterior pituitary gland, not, it will not be binding to hypothalamus. Next is when it binds to the anterior pituitary gland or adenohypophysis, then only the secretion of follicular stimulating hormone will be inhibited. Okay, so that is one thing. Again, when estrogen secretion happens, okay, estrogen will go and bind to both. It will be able to bind to this also and bind to this also. Okay, low level of estrogen will have negative here and negative here, means it will lead to negative inhibition of FSH and LH, and also inhibition at hypothalamus to secrete GnRH, means it will try to plummet or reduce or lower the secretion of gonadotropin releasing hormone, also FSH, also LH. Okay, but this inhibition is very less because these hormones are produced in very low quantity. Okay, so it consequently results in continued secretion of FSH and LH because the inhibition is quite less. Therefore, you have follicular growth stimulation, estrogen production, and thereby estrogen would go and bind to the, you can say, the stratum bacillus and causes the formation of stratum functionalis, and a lot of vascularization also would take place due to increased secretion of, you can say, vascular endothelial growth factor or VEGF. So you will have excessive vascularization at the endometrial lining. Okay, this is the specificity regarding this thing, and this entire axis is known as HPG axis. You might have read this in class 11th. If not, I'm repeating it for you. HPG is hypothalamus So this type of things that are directly asked in NEET examination, like HPA, HPG, okay? HPG is hypothalamus, pituitary, okay, gonadal axis, okay, means it pertains to the involvement of, you can say, the testes, the ovary, right, the gonads, right, in males and females both. You have HPA axis, hypothalamus, pituitary, adrenal axis, means the adrenal glands. Like that, you have multiple axes, okay. So what we are concentrating on is on the HPG axis here, okay. So this is how the HPG axis works. So only one dominant follicle, I told you, will be able to reach that size of 22, you can say 25, you can say, millimeter in size, and that will be able to attain maturity from both the ovarian follicles, from both the ovaries combined. And then as estrogen level rises, okay, as estrogen level rises in the, you can say, ovulatory phase, this will turn on, or this will be converted into positive feedback loop. I'll just show you how this will become a positive feedback loop, okay. So the thing is, before going on to that, I'll just tell you some of the important functions of human placental lactogen. Okay, and then you can say also the human chorionic gonadotropin. So we know that the human placental lactogen, okay, along with prolactin, human placental lactogen, along with prolactin, okay, and human growth hormone HGH, okay, they form a family of proteins with striking homologies in chemical and biological properties, okay. All these three hormones, okay, all these three hormones have a molecular mass in the range of 22 to 24 kilodalton, protein hormones only, okay, and they exhibit, you can say, both lactogenic and somatotropic properties. In this case, the human placental lactogen and also the human chorionic somatotropin, or HCS, you can call it somatomammotrophine, okay, HPL and HCS. There are two hormones, human placental lactogen and human, you can call it chorionic gonadotropin, or especially you can call it human chorionic somatomammotrophine also. They are both secreted from where? From the syncytial, or you can say syncytial trophoblast cells of the placenta. Okay, syncytial trophoblast cells that we had talked about in previous classes, present in the placenta only. Okay, so these same cells only, they also secrete the HCG that you might have heard of, right? Human chorionic gonadotrophin. Okay. That is why placenta is known as an endocrine organ. Okay, human chorionic gonadotropin, HCG, HCS, which is the human chorionic somatomammotrophine, and also human placental lactogen. So that is there. Now, what happens is human prolactin also is secreted during pregnancy and is synthesized and secreted by both the anterior pituitary gland and also the decidual cell that I told you. It forms the barrier between the embryo and the body. So the decidual cells and the, you can say, human, sorry, the anterior pituitary gland, they both produce, you can say, the prolactin also. So HPL, what it does is this hormone breaks down fats. Okay, so if you talk about the function of HPL, okay, it breaks down fats, means catabolism of fatty acids, which comes from the mother to provide fuel for the growing baby. Okay, for the growing baby. So that's what it does. Okay, so that is how this takes place. Now HCG, if you see, if you look at the HCG, okay, so you might have uh, heard about pregnancy kit tests. Okay, you might have heard of pregnancy kit test. So in that pregnancy kit test, what is present? Which hormone is present? Okay, so the hormone is human chorionic gonadotropin only. Okay, so HCG ensures that the corpus luteum, which is the temporary endocrine gland that takes the delta 4 pathway I had shown you for the, you can say, formation of progesterone. Okay, the temporary endocrine gland is the corpus luteum. Okay, that continues to produce progesterone during the first trimester of pregnancy. Okay, that is the specific function of, you can say, HCG and the HCG related molecules. Okay, this is the functions that are not directly present, okay, in the book, but are asked in the, you can say, comparative examination. So HCG is a term referring to four independent molecules, okay, each produced by separate cells and each having completely separate function. Okay, so there are specific HCG produced by the HCG produced by, you can say, villus or villi produce or villi having, villi possessing, you can say, villus syncytio trophoblast cells. Okay, syncytio trophoblast cells. Okay, that is there. Next is, you have hyperglycosylated, means too much glycosylation in HCG. That form of HCG which is produced by cytotrophoblast cells. Okay. Then hyperglycosylated means there is too many N-linked or O-linked attachment of glucose residues to this hormone. That is known as hyperglycosylation. So hyper, that is a post-translation modification, glycosylated HCG produced by cytotrophoblast cells. Cytotrophoblast cells. Then you have a pituitary made or SCG by the gonadotropic cells of the anterior pituitary gland. That is one type of SCG from the anterior pituitary gland. Just giving a perspective of where you, because HCG just doesn't mean you have one HCG only. Okay, One is this also, and one is the only the beta subunit, okay, made by, you can say, in case of malignant trophoblastic cells, okay, only the beta subunit of HCG made by malignant trophoblast cells in case of some cancers, okay. So there are four types of SCG, okay. So SCG promotes progesterone production by corpus luteal cells. So you can write down the functions of SCG now, okay. Functions of human chorionic gonadotrophin, okay. So what it does is promotes progesterone function, or promotes progesterone production by delta 4 pathway that I had explained to you. Production by corpus luteal cells, okay. Then promotes angiogenesis, okay. More and more angiogenesis means progesterone causes the formation of angiogenesis, means more artery formation. But HCG also, very few people know this, HCG also promotes angiogenesis. Angiogenesis is a term where it means more and more artery formation, or arteriogenesis, or just vascularity, too much vascularization. So it does promote angiogenesis, because angiogenesis is also a hallmark of malignant cancer, because when it is malignant, it is breaking down from one part, it is getting shifted and relocated in another body part. It would need blood supply, otherwise it will die. So it then causes the production of more factors like VEGF, vascular endothelial growth factor, to make more and more arteries. That's the function of cancer cells in accelerated angiogenesis. So this also, HCG promotes angiogenesis in uterine vasculature, which is very, very essential, okay, for the proliferation of the endometrial lining, right, in uterine vasculature. So not only does estrogen and progesterone cause angiogenesis, this HCG also supports angiogenesis, okay, and it promotes the fusion of cytotrophoblast cell and their differentiation also to make syncytiotrophoblast cells, okay. One other great function, promotes the fusion of cytotrophoblast cells, okay, and differentiation of differentiation to make, you can say, syncytiotrophoblast cells, trophoblast cells. So that is the ultimate, you can say, one of the ultimate functions of HCG, okay. And also it causes the blockage of any, you can say, macrophage or immune cell by, or present in the mother on the placental cells, because placental cells are also foreign, because they are produced from trophoblast cells, okay, they are also foreign. So again you must think of the fact that not only the embryo but the placenta also needs to be, you can say, protected from the own autoantibody attack, right. So it also blocks immune cell or macrophages from destroying the foreign placenta. Okay, or placental cells. Okay. One more great function it is it promotes, or you can say it suppresses. Okay, it suppresses any kind of myometrial contractions, means the muscular layer contractions during the course of pregnancy. Okay, suppresses any myometrial contraction during the course of pregnancy, because that could be very very lethal, right? So it suppresses any myometrial contractions during the course of pregnancy. Then one more great function is it causes the growth and differentiation of the umbilical cord. Growth and differentiation of the umbilical cord. Okay. Seventh point is, you can say it signals the endometrium about the, you can say, forthcoming implantation, makes it ready. Okay, signals the endometrium, keeps it ready for implantation. So these are the penultimate functions which are perpetuated by human chorionic gonadotropin. Okay, and you will say, what is the use of hyperglycosylated HCG? That functions to promote growth of cytotrophoblast cells and invasion by these cells. Okay, which occurs in implantation of pregnancy. So that is what it does. Okay, so these are the great functions which are perpetuated by human chorionic gonadotropin hormone, okay, which everyone should be aware of. Okay, so in the next part I'll tell you about the ovulatory phase. Okay, before jumping on to the ovulatory phase, okay, so we have completed the follicular phase completely. Okay, so the thing is, I'll just show you, okay, one diagram, okay, one great diagram that comes and that is asked in the, you can say, exam, board examination. So I'm just sharing my screen. Okay. Now you'll be able to see this diagram in just a few seconds. Good evening, Tarun. Okay, so you'll be able to see it. Yeah, now you can see the diagram specifically. Just look at this diagram. There are two graphs, okay, FSH and LH, okay, that is in milli international units per ml. Okay, so this is the FSH graph. You can see LH surge happening at day 14. You can see that LH surge, which is extremely required, or without that ovulation would be indispensable. Okay, multiple times I have told you that the estradiol which is produced by the granulosa cells will then cause the, you can say, expression of the LH receptors on the granulosa cells itself, on the follicular granulosa cells itself, and then that will lead to ovulation via the signaling cascade downstream. And then these are the days of ovulation or ovulatory cycle, or sorry, the menstrual cycle, and then this is progesterone in nanogram per ml. Okay, so you can see the progesterone level peaking when corpus luteum has been produced, okay, and then again disintegrating when corpus luteum becomes again corpus albicans if there is no fertilization, okay. Estradiol level peaks when there is Graafian follicle, more and more estradiol level, more and more estradiol is being secreted by the, you can say, granulosa cells, okay. 17 beta estradiol, this is disintegrating corpus luteum from the previous pregnancy. Now this is the, you can see the follicular phase, this is ovulation, estradiol level has dropped, and now after ovulation you can see this peaking, and this is the, again this is the same thing, same graph here, okay. You can see 21 to 28.1, 21 to 28.1, okay. This is the preceding graph here. So this is how you analyze, okay. Now this, both these graphs, all these four lines, or they can be also, you can say, superimposed in some books, okay, but the units will not be, but sometimes they do superimpose it, because this is milli international units per ml, and that is nanograms per ml. They should not be, but sometimes they are, okay. There are some special quality people who do this. So what you should know is, you should concentrate on knowing the specific graph and be able to comprehend the entire menstrual cycle, the various phases of the menstrual cycle, and correlate it with the upregulation or downregulation of the various hormones like FSH, LH, estradiol, and progesterone, as per the current menstrual cycle which is going on. Okay, sometimes they will not give you all this. You have to name all these things, or you have to write as per the graph what is happening, or they'll just draw the graph and not let, or not label the hormones. They'll just erase this one, LH, FSH, estradiol, and progesterone. You have to identify which graph or which peak represents, or which line represents which hormone, okay. That may be a 4 mark question, okay. Just one hormone's name you have to write, and that will get you 1 mark in your board examination, or you will be awarded 1 mark for that. That will fetch you 1 mark, right? So be very very cautious regarding it, okay. That is what my message is, that how to pretend, sorry, how to comprehend and be able to, you can say, solve the graph, okay. That is the main basics of, you can say, this specific exercise, right? Knowing the graph, okay, being able to comprehend and solve the graph. So next step is, now we have the ovulatory phase. So we know that LH surge does take place during the ovulatory phase, day 14, right? Ovulatory phase or day 14. So LH surge does take place, and when LH surge does take place, what will happen is, you will be able to see ovulation. So in response to LH surge, you can say the follicle ruptures and the mature oocyte is assisted to the fallopian tube by the fimbriae. The fimbriae would catch that, right? And there it remains viable for fertilization for around 24 hours. Now, following ovulation, the follicle remains luteinized. It secretes estrogen and now also the progesterone, that is the corpus luteum. Okay, so following ovulation, the follicle remains luteinized. Okay, it secretes estrogen and also progesterone. And also progesterone. So Then what happens is, I'll draw the negative feedback loop for you. Hypothalamus. Here you will see the negative feedback becoming positive feedback. So here hypothalamus, then you have anterior pituitary, adenohypophysis. Okay, and then this will be secreting your gonadotropin releasing hormone. Okay, and then here I can write ovaries and follicles. Here I can write uterus endometrium. Endometrium. Now just see the specific pathway. Okay, so hypothalamus GnRH positive here. Okay, and is the effect is positive. So FSH will come and bind. Okay, again effect is positive. You can say LH would also come and bind, right? Now, ovaries are making estradiol, but the estradiol level has raised or not. It has raised with in comparison to the follicular phase. So increased estradiol level here. Okay, increased estradiol level needs to, you can say, increased estradiol level leads to the formation, or you can say the change here. This is a very dramatic change. The change or the switch from the negative feedback to the positive feedback. Okay, to the negative feedback to the positive feedback, meaning this one, estradiol in high amount is being secreted, that will act as positive and will perpetuate hypothalamus to secrete more GnRH. If you compare it with the negative feedback loop that I drew for the follicular phase, the estradiol was acting as a negative regulator of hypothalamus and was inhibiting or prohibiting hypothalamus from secreting GnRH and also pituitary gland, right? But here it is not binding to anterior pituitary part 1, and part 2 is it is causing the hypothalamus to secrete more and more GnRH, that then it will proliferate anterior pituitary, or you can say stimulate anterior pituitary to secrete more and more FSH and LH, and then this is known as LH surge. More and more LH will bind to the granulosa cells as well, because the granulosa cells here have also expressed LH receptors. Okay, it is very important to know. They are also expressing LH receptors, as 17 beta estradiol has bound to granulosa cells, has caused downstream signaling, because that is again an steroid hormone. It has bound to the nuclear receptor present in the granulosa cells and has caused the expression of LH receptors. LH is a protein hormone. It will bind to the surface Okay, protein receptor on the surface, the protein coupled receptor or GPCR present on the LH is present on the granulosa cell or follicular cell receptor, sorry, or follicular cell plasma membrane or surface. Okay, so this is known as positive feedback here, means this estradiol is causing more and more GnRH to be secreted from the hypothalamus. This is the positive feedback loop. And this estradiol, 17 beta estradiol, when it binds to the uterus, okay, it also causes the endometrium to thicken. Okay, so growing follicles, if you see, if I write it here, growing follicles, they begin to produce high levels of estradiol, which will, you can say, stimulates stimulates GnRH secretion, GnRH secretion by the hypothalamus, by the hypothalamus. Okay, in response to this, what will happen? The LH and FSH levels will also rise. FSH levels also rise, right? And that results in ovulation about a day later, okay, on 14th day. And it will break the diplotin arrest also. Keep that in mind too, okay? So LH search will cause the breaking of the diplotin arrest also, right? The CGMP will not be able to come through the gap junctions, that will be closed, okay? And then MPF, the mitotic promoting factor or the maturation promoting factor, will not be inhibited, okay? And how it will not be inhibited, since your CGMP cannot come in, so phosphodiesterase 3A will not be inhibited, and that will, phosphodiesterase 3A will break down cyclic AMP to 5 prime, you can say AMP, and then cyclic AMP cannot in turn further inhibit the maturation promoting factor, and that will lead to the, you can say, evasion from the diplotin arrest, okay, release of diplotin arrest. And so all these things are extremely interrelated. This entire chapter is very, very interrelated if you think like that, right? From the conceptual level. So resulting LH and FSH levels will also rise here. Then you can write resulting in ovulation about a day later. Resulting in ovulation about a day later. So here you should be taking this pathway into consideration. Gap junction is closed, so CGMP cannot enter, right? If you have forgotten, cannot enter the granulosa cells. Now, if that they cannot enter, CGMP was inhibiting phosphodiesterase 3A. Now, no inhibition. If no inhibition, you can say CAMP will be broken down by phosphodiesterase 3A to 5 prime AMP, and CAMP was inhibiting the MPF, right? Maturation promoting factor. Now this cannot inhibit maturation promoting factor. Inhibit MPF. So diplotin arrest is broken, right? Very important. It is a fundamental pathway that is occurring here, right? Now more and more LH and FSH secretion, more and more estradiol production, and binding of more and more estradiol to the uterus and endometrium will also cause angiogenesis. Okay, so it will cause the endometrium to thicken as well. Cause the endometrium to thicken. So this is a very important, you can say significant negative feedback, sorry, positive feedback loop here, right? Very significant positive feedback loop that perpetuates. You can see here. This one. Okay. So here the positivity occurs because the switch occurs due to what? Due to estradiol. Estradiol becoming more and more in concentration, its concentration increasing by leaps and bounds. That causes the, you can say, signaling event because it's an estrogen hormone, sorry, it's a steroid hormone. It binds to the nuclear receptor in hypothalamus, and then it will cause the transcriptional changes which will lead to increase in the concentration or increase in the secretion of GnRH instead of decrease, right? Stimulation of GnRH secretion, and that will lead to the ovulatory event, right? That leads to the ovulation, and because of that only you can see the diplotin arrest and all those things, the overcoming of the MPF, diplotin arrest, evasion, and then ultimately the ovulation would take place. Okay, so after estradiol reaches moderately high levels in the mid follicular phase, okay, we saw that it exerts negative feedback on GnRH secretion and FHS levels begin to decline. Okay, and human ovarian follicles, which I told you, in the follicular phase only, they also contain inhibin, which inhibits only FSH but not LH. Okay, and we know that there is a related ovarian peptide also known as activin that stimulates FSH secretion. Okay, the ovary also produces one more hormone. Okay, so there is another hormone known as the another peptide known as activin, which stimulates, you can say, FSH secretion, works opposite to that of inhibin. Okay, FSH secretion. That is there. One more thing I'll tell you is the ovary also produces something known as follistatin. Okay, very important. Follistatin. Now, what does follistatin do is it is a glycoprotein. Glyco means glycosylated. Glycosylation occurs N-linked and O-linked in the Golgi apparatus post-translational modification. It is a glycoprotein. Okay. What it does is it inhibits FSH secretion. How? By binding to activin and neutralizing its effects. Very essential. It binds with activin. Now binds to activin And we know that activin stimulates FSH secretion, but follistatin, after binding to activin, it neutralizes activin. Means now it can no longer stimulate FSH secretion. So what is the consequence, or what is the cumulative effect? The real effect is follistatin is able to, you can say, inhibit FSH secretion, because now there is nothing which is able to counteract the, you can say, effects, or the, you can say, the ramifications or the consequences, or any kind of trigger which is perpetuated by inhibin. Okay, so it inhibits FSH secretion from that perspective, because there is nothing which will hold back inhibin now, right? So you can say that in the last days of the follicular phase, estradiol levels are very, very high, okay, secreted from several large follicles in each ovary. And now they exert a positive feedback on GnRH, gonadotropin releasing hormone secretion. Okay, now this increase in GnRH, it causes LH surge on day 14, because we have multiple large follicles in each ovary. Right? But only one would reach a diameter of 20 to 25 mm, and one would ovulate. So multiple large follicles, large tertiary follicles in each ovary, that would release a lot of, you can say, 17 beta estradiol, and that high a concentration of estradiol would act as a positive feedback for GnRH secretion by hypothalamus in the human brain. Okay, so now does all the specificities they change. Okay, and then this increase in GnRH causes LH surge on day 14. The protein arrest broken, and then you can say ovulation. Now GnRH along with FSH and LH, because that will induce FSH and LH production. So the pulses, because I told you this is a pulsatile secretion, means intermittent secretion, stops again starts, it's not continuous, right? So pulsatile secretion increases 10 times in amplitude, means with each, you can say, secreting event, 10 times the hormones are secreted compared to the normal amount. So the GnRH pulses increase 10 times in amplitude and occur every 30 minutes during the LH surge, means every 30 minutes that will repeat, means more and more and more and more of GnRH, that will lead to more and more of FSH and LH release during the LH surge. So the frequency also increases and the amplitude also increases. Amplitude means the level of the hormones secreted with each, you can say, GnRH secreting event or GnRH release from the hypothalamus, right? So I hope you are able to comprehend the lecture. If you've had any doubts, you can clarify your doubts with me on my personal number. I'll be replying as soon as possible. Okay, in the next class we will be addressing the luteal phase with again negative feedback loop, because that will again revert to negative feedback during the luteal phase of the menstrual cycle. Okay, thanks a lot. Let's meet you all in the